I'm going to start by providing a little bit of a disclaimer. So I'm a geneticist and a developmental biologist. In particular, I'm interested in how you put together a nervous system. And this field of autism involves so many different disciplines to understand all the different problems: immunology, epidemiology, statistics, neurology, neuroscience, cell biology. And in a few of those, a small subset of those, I feel like I'm a fairly educated person. But of course, a lot of the issues that you would like to hear about are outside of that area of expertise. And what I'm going to try and do is be a reasonably informed interpreter of that data. So I'm going to tell you, I'm going to try and address some of those issues. I actually want to thank Joel and Ellen for this. I think it's incredibly important to have this kind of exchange. And I thank them for the opportunity, and I thank them for their service to provide this kind of information out there. And I'm a big believer that you do not solve these problems by avoiding them. And those of us, quote unquote, in the ivory tower need to get out more. And this is a mechanism of doing so. So I'm going to try and cover quite a few topics today. And I'm going to try and give you my best interpretation of what is out there and how you can go get more information for yourself. Okay, so let me give you a little bit of a history first. This is actually a set of disorders that was described some time ago by Leo Kanner in 1943, describing 11 children in his first study that had what he described as social isolation. Very shortly thereafter, Hans Asperger described another set of children that he described of having this autistic psychopathy. Now, there are really three major phenotypes or changes that characterize these children. And my colleague, Dr. Reif, will tell you much more about that than I will this morning. But just to begin, they're impaired social development, changes in language development, and rigid and repetitive behaviors. Now, we also know that there's a very difference in the male to female ratio, about 4-to-1. And here's probably the most important point I want to make from this intro. We know now that the prevalence is about 1 in 150 for autism spectrum disorders. And this is good data. This comes from a recent report of the Autism and Developmental Disabilities Monitoring Network back in February of last year. And this is data from 14 sites across the United States. And this is collation of a variety of different information from state health departments, et cetera. So whether or not you believe that there's an actual increase in the incidence of this or not, this is a real number that you can rely on. 1 in 150. That's a huge problem, obviously. Let's compare that, for example, to childhood cancer. Between birth and 18, the incidence of childhood cancer is 1 in 800. Okay. So this is a very big number, and the truth is we do not understand this set of disorders very well, not nearly as well as we understand childhood cancers. So we have a long ways to go, and it is a big problem, and we better get moving. So there are a lot of features associated with this disorder, and that's one of the things that makes it so hard to study. In about half of the patients that have a diagnosis, they have also a diagnosis of mental retardation. Seizures is quite common among these children and into adulthood. About 20% of these kids have macrocephaly, which is just a medical falutin word for a big head. They have postural and motor abnormalities. They can be either less sensitive or hypersensitive to stimuli. So it's not just that triad of things. It's a very complex presentation. They can have eating, drinking, or sleeping changes as well. There's a high frequency of gastrointestinal complaints from the families and these children. Mood changes, self-injurious behavior. In other words, it is incredibly complicated. Now, in addition to that complication, this set of disorders overlaps with other quote-unquote defined disorders. And it sort of begs the question, well, are they distinct? Are they totally separate entities? What are they? And of course, this is a common problem for any disease that affects behavior. The same is true for schizophrenia and depression, schizoaffective disorder, etc., right? assessing behavior and putting it in nice, neat little boxes is almost impossible. And it's true for adult psychiatric disorders. It's true for behavioral disorders in children as well. So there are at least 28, and it's, since I compiled this number, there's probably another 10 of known genetic disorders that have overlap with autism. Tuberous sclerosis complex is one. This is a dominant genetic disorder that has about 90% of these children have seizures, and somewhere between a quarter and a half of them get a diagnosis of autism spectrum disorder as well. Angelman-Prader-Willi syndrome is a chromosomal anomaly on chromosome 15. Fragile X, about a third of those children get a diagnosis of autism. Are these distinct disorders? Are they autism? Are they models for autism? Those are very readily debatable questions among the people that study them. Down syndrome, neurofibromatosis. And I'm going to talk a little bit more about this disorder in a few minutes. So, autism is complicated and extremely heterogeneous. And it can be difficult to diagnose with the same reliability. This is not a disorder that, let's say, a well-informed person in a school can say, aha, this child has this. It's difficult, and there are different criteria for diagnosing it. There are quote-unquote research-level diagnostic measures, and we have some experts at the University of Minnesota that work on those and use those to assess the children that come to see us. But suffice it to say that it is not trivial to reliably make the same ascertainment. And this is a very important question. I happen to think it's probably 30 disorders all rolled into one. It's not going to be, in my view, a distinct entity. And I'll tell you a little bit why I believe that in a moment. But because behavior is so hard to measure, we're lumping, because we don't have the tools to accurately segregate them into different distinct groups that we can all agree on. So that's a problem, and it makes it harder to study. Okay, so I'm going to give you an historical example of this issue of how many disorders are embedded there. And this is, the example I'm going to give you is a syndrome called Rett Syndrome. One of the first things that you see in this, one of the characteristics of children that have this genetic syndrome, is loss of purposeful use of the hands. And they also have slowed brain and head growth, gait abnormalities, seizures, mental retardation. It's in girls almost exclusively. It turns out to be the mutation is on the X chromosome. And these girls can exhibit autistic-like behaviors in the early stages. In fact, they were diagnosed as autistic before we knew a lot about this disorder. Now, as it turns out, a geneticist at Baylor named Huda Zogby went after this disorder and over a period of about 15 years identified the specific genetic change that was responsible for this disorder. And it was caused by mutations in a gene called MECP2. It's an individual gene on the X chromosome. Now, what has really happened is that once that genetic test was available and you could determine, is this something else or is it Rett syndrome, people began to say, aha, now that we have this DNA test that's very reliable, they began to say, okay, we see the difference. These children have the following differences. And so it's now really not a part of autism spectrum disorders. It's Rett syndrome, because we now have a better way of assessing it. And once we had that measure, people began to notice the distinct behavioral characteristics that would help them identify it. Now, what does this gene do? Well, it's a very interesting genetic story, and it encodes a protein that's involved in controlling gene expression. That is, it controls the genetic information that we have and how that's expressed. And it turns out to be a protein that recognizes a specific type of modification of DNA, and you're going to have to forgive me. I'm going to jump into some molecular biology, and I hope I will carry with me more than 30%. We'll see how I do, and you can wave me off if I'm really messing up. So I'm going to start with what I call, what is the molecular logic of living systems? And here it is in a nutshell, right? The set of instructions is in your DNA. Now, a liver cell expresses a different set of genes than a neuron or a kidney cell or a skin cell. And so there's differential control of that information. And how does that happen? Well, step one is the DNA, which is, of course, in a chromosome. It's quite an amazing thing, right? I mean, a chromosome is a single DNA molecule. It's a humongous DNA molecule. One molecule is a chromosome. And that information is sitting there in the nucleus. How does that get out? How is that expressed to actually change what the cell does? Well, the cell makes a copy of it, an RNA copy that leaves the nucleus and goes into the cytoplasm of the cell. And there, it's translated into a protein. So these are the workhorses of the cell. They encode for structural components of the cell, receptors on the cell surface to see the environment, signaling molecules. So the way this whole, and this is polymer chemistry, right? This is a polymer, this is a polymer, this is a polymer. How this all works is you make a copy of that information. It gets to the machinery of the cell that converts it to the actual entity, the protein, which does the job of the cell. Okay. Now, so the first step in information flow is getting a copy of that into the cytoplasm where that machinery can read it. And translation, which is the process of taking the RNA and making a protein of it, is the critical step in getting that information expressed. Okay, and these proteins perform many essential cell functions. They're enzymes, they're involved in signaling, they're structural components of cells, they're receptors, etc. Okay. Now, how does this all fit into Rett syndrome? Well, DNA is packaged into chromosomes. And it's not just, it's actually quite a challenge because DNA in any nucleus, it's about 2 meters of DNA. If you stretch it out, it's 2 meters. And it's got to fit into a teeny, weeny, weeny little nucleus. It's like putting 24 miles of string into a tennis ball. All right? So there's a lot of packaging that has to go on. So what does it look like? Well, here's a chromosome that's been condensed, at the stage when the cell's about ready to divide. And this copy of the chromosome is going to go to one cell, and this copy is going to go to the other. Now, if you look at this material when a cell's not dividing, what's it look like when it's doing its thing? Well, it's a bad day in the spaghetti factory, I can tell you that much. It's a mess. Well, it's actually, of course, not a mess. The cell knows what it's doing. And it is packaged. And it's packaged in a way that information can be accessed and turned off appropriately. In fact, here's what it actually looks like if you look at it under a very high electron microscope. Literally, so this is the DNA here. This little strand is actually a single DNA strand. And you see these little balls on it. And this is part of the packaging structure. It's called a nucleosome. Okay? And this is after you've treated the DNA to get rid of some components so it begins to open up a bit. And if you don't do that, you see that this structure looks like this. So there's a level of compaction here. What I'm trying to tell you is there's a whole system of enzymes and proteins that control the level of compaction throughout the cell cycle and making sure information is obtained appropriately. As it turns out, the protein encoded by that MECP2 gene involves chromatin compaction. All right? So one of the things is what it does is it turns out it recognizes a particular change in DNA, a methylation, and this methylation instructs the cell, clamp down on this DNA, shut it off, package it up, make it inaccessible, turn off what's going on there. Okay? That's what that methylation signal does. And MECP2 recognizes that methylation. So it is part and parcel. So here's what, the beads on a string, right? Here's the DNA going zippity-zippity around these little nucleosomes. And there are other proteins that package this. And here's a little methyl group stuck onto the DNA. And these proteins that recognize that methyl group, they're involved in compacting it and turning off gene expression. And that's what MECP2 does. It recognizes this, and it's involved in condensing chromatin down to a state where the underlying genes cannot be expressed. Okay? So, just to add one level of complexity to this, so we know that a mutation in a specific gene causes this syndrome. But interestingly, it's not actually inherited. You go, well, wait a second, I thought you just said this is a genetic disease. Well, it's genetic in that a change in the DNA is responsible, but that change occurs in the individual who's affected. That girl does not get the mutation from her parents. Okay? All right, now here is what this area of research has opened. Right? And the question then becomes, okay, swell. What are the genes that are expressed in the absence of MECP2 that are normally shut off, right? That's the issue. And that's where that area of science is going. Now, and I'm going to come back to this. I'm going to try and weave this together. We'll see how I do. So let me come back a bit to, the $64 billion question. What are the causes? Genes or environment? That's a simple place to begin. What is it? And I often hear in the media and things like that that these are provided as alternatives that are mutually exclusive. One of the most important things I can tell you is they are not mutually exclusive. In fact, almost all complex diseases are a combination of what you bring to the table with your genetics and what you see in your environment. Okay, so let me give you an example. And this is not going to be autism. This is going to be obesity in Pima Indians. Because it's a great example of this relationship between genes and environment. So I used to work in Arizona. So I got to meet some of these folks. So this is a picture of the Pima Indians in 1886. And they served as the scouts for the cavalry who were running around chasing other groups of Indians that didn't want to toe the line. Now this group of people lived in southern Arizona for 2,000 years. And I can tell you, they were lean and mean, if you know what I'm saying, right? Well, when they became centralized onto reservations, and they acquired the diet that was given to them by the Bureau of Indian Affairs, they developed the highest rate of obesity and adult onset diabetes in the world. So this is an example of how, they didn't have that before the cavalry came, I can tell you that much. So what happened here? Well, they had a genetic susceptibility, and then they came onto this Western diet, and boom, they had this disease. And this same principle can be applied to cardiovascular disease, type 2 diabetes. I mean, it's in just about every complex disorder, it's this interplay that's important. Okay. So what I'm really saying is that to understand, how heredity might contribute to disease, it's not a straight line, right? It's not a straight line. It goes through the environment. So it's this interplay that's really critical. And that, of course, makes it a more complicated problem because you're dealing with interactions of things. Okay, so with that, is there a consensus, for example, that ASD is on the rise and hence environment is playing an important role? Obviously, if there's a real increase in the incidence, our genetics isn't changing all that much from decade to decade. So if there is an increase in one particular region of the country, that says environment is playing an important role. And of course, it's even more important because that is something we can control, right? So it's a very essential question. So is there consensus that this is happening? Guess what? No, there isn't consensus. And I'm not an epidemiologist, but let me give you an idea of the flavor of what's out there. Well, one of our colleagues at Mayo, Bill Barbarisi, published a paper, I believe about a year ago or 18 months ago, that said that for Olmsted County, where they have terrific records, they found no evidence that there was a true increase in the incidence. They felt they could account for the change by virtue of recognition of this disorder, change in diagnostic criteria. One of my colleagues at the Mind Institute at UC Davis in California is publishing a paper in December in the journal Epidemiology, and she's a terrific epidemiologist. Her name is Irva Hirsch-Pachotto. She has looked at the California data, and she argues that you cannot account for the increase that they've observed over the last decade simply by changes in reporting and diagnostic criteria. So who's right? Well, maybe one is right for Olmsted County and another is right for California. The European groups, for the most part, say we don't see this rise in prevalence. So the bottom line is there is no net consensus on this. It depends on the study, the geography of the study, etc. But it doesn't mean that environment doesn't play a role, right? If you don't find something, it doesn't say it is there. It says you haven't found it. And that's a very important distinction, right? Every study, every method has its limitations. And Joel in his comments, talked about this. What is the sensitivity of what you're doing? What can you pick up if it was there with the power of your tools? And that's important to bear in mind. Okay, so what do you, I mean, obviously this is an important and controversial environmental contributor that parents have been worried about for more than a decade. So what about this? Is it an environmental contributor? And I'm going to, since I'm not an epidemiologist, I'm going to defer to this report. And the last one that was comprehensive came out of the Institutes of Medicine of the National Academy. So this is a group of people that are our country's best. And they were asked to review, I think, 20-some different studies, some in the United States, some internationally, about, well, what is the deal with vaccines and autism? And here's what they said. The committee concludes that the body of epidemiological evidence favors rejection of a causal relationship between the MMR vaccine and autism. The committee also concludes that the body of epidemiological evidence favors rejection of a causal relationship between thimerosal-containing vaccines and autism. So that is what the best people we've got have said. And my response to that is, that's as good as I can go. You know, I think it is important to always be skeptical of who's saying what, etc. But I can reassure you that 99% or more of the people that are studying this problem, like me, we have no axe to grind. We have no money from companies. We are motivated, part of the motivation that we get to be professors, which is a privilege, is service. Okay? So the vast majority of people that are studying this problem do not have an axe to grind. They're just trying to figure out, make a contribution. So that's what our best panel of experts said as of 2004. If you want more information, I encourage you to go to the CDC, the Center for Disease Control website. They have press releases there that are very informative. You can see what were the answers from Tom Insel, the director of the National Institute of Mental Health, to various reporters saying, well, what's this business about mitochondrial disorders and the susceptibility of children with mitochondrial disorders and vaccines? And you can read that discourse. It's all up on the website, and I encourage you to do that. Okay, so I want to go back to this gene-environment interaction a bit and some more lessons from Rett syndrome and models of this syndrome in the mouse. It's truly Huda Zogby, this investigator in Baylor. So she identified the gene in humans and said, okay, to understand this, we've got to make a model. So she made a model in the mouse. And one of the most amazing videos I've ever seen is when she showed a video of a young girl with Rett syndrome walking down the hall. And to display one of these characteristic motor changes. And one of them is sort of a hand-wringing behavior. This girl walked with a little bit of a shuffle and had hand-wringing behavior, sort of like that. And then Huda showed this video of a mouse model, so knocking out the same gene in a mouse. And she said, you know, initially, and I should say knocking it out was not lethal or anything. These mice walked around, etc., mated, had normal reproductive capacity. And she said, initially we thought we were going to have to actually do a molecular test to determine was this the mouse we wanted each time when we bred them. But she said we learned we didn't have to do that because we just had to pick them up. Now, if you take a wild-type mouse and you pick it up by the tail, you can imagine what it does. It goes, right? It goes, give me something to hang on to. All four limbs shoot out looking for something to grab onto. Guess what happens when you pick up this Rett syndrome mouse? Amazing, right? Amazing reproduction, if you will, of that motor change. And so you don't have to genotype anymore, just pick them up, and if it has the mutation in the mouse, it does this paw-wringing behavior. Now, the severity of that phenotype in the mouse or in these girls is determined by the level of DNA methylation, right? There's two partners here, the protein that recognizes that methylation and the methylation itself. Now, this is particularly interesting because DNA methylation is sensitive to dietary factors. We know this, right? There are a whole bunch of things in the diet that influence your capability to put this particular modification in DNA. Things like folate, vitamins B2, B6, B12, etc. In fact, you can moderate the severity of the problem in those mice by changing the diet of these items. So here's a pure case that says, oh yeah, genes and environment, they interact very clearly. So I think that's very instructive. So I'm going to turn to another syndrome that also allows us to talk a little bit about this relationship between genes and the environment, and that's tuberous sclerosis. I listed this earlier as one of the disorders that, half of these children get a diagnosis of autism. So it's a dominant genetic disorder. About 90% of these children have seizures, and about 50% of them receive a diagnosis of autism and also ADHD. It's caused by mutations in one of two genes. So it's pretty simple genetically. And they're called tuberous sclerosis complex 1 or 2. Now, as it turns out, these genes also control growth. In fact, they're inhibitory on growth. All right? And in fact, if you have mutations in these kids have benign tumors. All right? Now, I'm going to come back to this as well. But as it turns out, this signaling system that involves these two genes and the proteins encoded by those genes is a critical regulator of growth and cell division in development in all organisms, everywhere. So that's good. That means we can model it in other places. And here is this pathway. All I want to convey to you here, this is a cartoon of the cell membrane. So this is the outside of the cell, the inside of the cell with all these cute little circles regulating events in the cell. And I just want to point out that it's a pretty complicated system, but central to it are these two genes. And the proteins of these two genes, they actually form partners in the cell. Okay? They do the same thing, and they do it together, which is why if you clobber either one of them, you're in trouble. Now, this pathway that regulates growth, and I mean it, so it determines how big you're going to be, how big your cells are. This pathway has many inputs. One of them, I'm getting ahead of myself. One of the questions is, how could this pathway that affects growth, what's it doing in nervous system development? I mean, these kids have seizures, et cetera. What's it doing there? Now, as it turns out, that pathway that I just described, which has nutritional inputs to it, it's conserved everywhere, including in, believe it or not, a fruit fly. Now you say, oh my God, I get this question a lot. How are you going to identify an autistic fruit fly? And of course, that question, bears answering. And the point is that if you're interested in understanding fundamentals of how a nervous system is put together, you can use a simple system like a fruit fly. And if we think of our system as maybe a Boeing 777, a fruit fly is a Cessna. But if you're going to try and understand an airplane by taking out a part at a time, you want to do that on a Cessna first. And then you extrapolate what you learn from those experiments with a Cessna to a 777. I can only imagine how many little chips you would take out and nothing really happens except if a goose hits the front of the airplane. I mean, who knows? So this pathway in all its representation is actually found in fruit flies. And it's interesting that this pathway is a critical regulator of growth. And there are two genes that serve as the brakes to this signaling system. That is, they impede growth. One of them is TSC, and one of them is a gene called PTEN, which is also a tumor suppressor. Very important in cancer biology. Now, what we're interested in is how do these genes affect susceptibility to autism? And as it turns out, remember I told you about 20% of the kids with autism have big heads? About 5% of those kids have a mutation in PTEN. So two different genes playing inhibitory roles on the same pathway have independently been shown to affect the development of autism. This is not a coincidence. So we're studying this system in about the simplest model we can, and that is the fruit fly. Now, one of the advantages of the fruit fly is you can really look at the pattern and organization of the nervous system. This is actually a fruit fly retina. And this is showing the projections of those neurons from the retina into the brain. And you can see they're very regular, which means if something goes wrong with those patterns, you can see it lickety-split, right? The other system that we study in fruit flies is that the fundamental unit of the nervous system, the synapse. How do two cells communicate with one another? Right? It's a chemical communication system. How does that happen? Well, fruit flies are more like human beings than you might suspect. Some of us are more like it than others. So you can study these events in a fruit fly. So let me show you what happens if we make mutations in those genes in a fruit fly that we know play some role in autism susceptibility. So what we're looking at here is the projections from photoreceptors coming into the brain of a fruit fly. And this is the pattern that you should see at two different stages in development. This is early in development, before all these processes have finished their trajectory, and this is later in development. And this is what happens when you disrupt TSC1 in a fruit fly. This is a mess. Okay? So those axons do not know where to stop. They go to the place where they should stop, and they just keep on barreling through and grow deeper parts of the brain. P10 mutations do the same, but not as severe. And it's kind of interesting that in terms of the incidence and severity, TSC mutations are much more prevalent in neurological disorders, including autism, than are P10 mutations. Okay. Now, we actually decided to see if we could create a model for tuberous sclerosis in a fruit fly. And so what we've done is disrupt this signaling system just in the neurons, just in the brain. And we've tested them for behavior. And this behavior is their ability to see light and run at it. I mean, as you know from anybody who does barbecuing out on their porch, bugs like to go to light. So do fruit flies. You can actually measure it. And so what we've done is make a fly, and we've been able to evaluate this behavior. And what we've shown here is the control. See if I can run it again. Nope. Okay, too late. We have a behavioral measure of this. And these flies that have disruption of the signaling system, they show this behavior much less well than a wild-type fly. And so that gives us an assay for determining what modulates this behavior. Now, I mentioned before that nutrient inputs are very important to the signaling system. So one of my undergraduate students had a great idea. Let's put these flies on a diet. And I can tell you that those deficits, the behavioral ones and the axon guidance one, we rescued them by limiting their calories by about 30%. I was astonished. Okay, now, you think about it. What is changing in our environment for our children? Well, I've been involved in raising 6 kids, and I can tell you, the longer I go to playgrounds, the bigger the kids get. Right? Our caloric load is very different than it was in 1957. Chewy bars weren't out then. Okay? So that is one possible environmental contributor that we should not discount. I mean, I talked about the things that affect methylation status, right? But also, this pathway is also affected by a nutrient environmental factor. Now, is this just some crazy nut guy working on fruit flies? No. There are dietary interventions that we know affect neurological disorders, namely seizure disorders. Back before we had medications to treat people with seizures, there was a diet that was largely implemented at Johns Hopkins called the ketogenic diet. And this was a diet which decreased amino acid, protein levels. Most of the calories that they were delivered were from fats, from lipids. And this has as an effective treatment of seizures as any of the modern medications that are used for epilepsy. Now, it's a very difficult treatment to impose, but it works. This, to me, is an amazing clinical fact. You can take someone with seizure and get as effective treatment with a dietary change than you can with some of the medications that have been screened and put on the market. So that's my way of saying it's not just us crazy geneticists that think this dietary role might be important. Okay, so I've danced around genetics quite a bit. Well, what about genetics? What is the evidence that this really is playing a significant role of susceptibility to this disorder? Well, probably the first fact is that the incidence in families is 50-fold higher than in the general population. It runs in families. And one of the ways that you can look at this is to look at twins and say, all right, are identical twins, if one has autism, does the other have autism, and how often? That's called concordance. So if one twin has it and the other does too, and they're concordant for it, they both have it, that's listed as, what is the percentage of that concordance? And it's very high, for autism. It's 60 to 80%. Now, we know that about 5% of the autism cases, you can find visible chromosomal abnormalities with them. We also know that more common in family members are reading and speech difficulties, anxiety disorders, etc. The male-female ratio is 4 to 1. That's genetics. But I want to emphasize that genetics does not exclude environmental factors. It's really this interplay that's going to be the most important. So what good is this? You say, all right, swell. You're going to get your little DNA test. And what is it going to do for my son? It's a good question. Well, my answers are, first of all, it will, in the end, allow us to improve diagnosis, just like it did for Rett syndrome. We could identify a subtype by that. It also allows us to identify people early, because we can get DNA very early. So if we have DNA tests, that would be very useful. So early diagnosis means early intervention. And the third point is mechanism. It allows us to understand mechanism. What is the basis of this? Now, this approach has been taken in cancer biology, and it's been very effective. But I will tell you, we are 25 years behind them in behavioral genetics. Okay? We have a long ways to go. Let me give you an example of the power of understanding mechanism. And this is for a tumor in the GI tract, where a bunch of investigators have identified the specific genetic lesion of those tumors, and they've identified a drug that specifically targets that genetic lesion. It's called Gleevec. This is a PET scan of glucose uptake, and this is the individual with this tumor, and here's the individual 1 month after his treatment. Amazingly effective drug. Very little side effects, very effective, because we understand the molecular basis of those tumors and can intervene. So what do we know about the genetic contributions to autism? Well, it's hard going. It is not simple Mendelian genetics. It's not like cystic fibrosis. It's not like sickle cell anemia. It's not like Huntington's disorder. It's not a single or a few genes that behave in a Mendelian way that account for it. We know that already. It doesn't work that way. But we are making progress. 10 to 20% of the children have identifiable genetic changes, and our methods are improving quickly. Our tools for recognizing genetic change are exploding. Well, what do we know about the genetic contributions? Well, a number of relatively rare disorders account for small percentages. Recently, we have come to understand that DNA copy number differences are very common in people. In other words, imagine the book of life, as Bill Clinton called the DNA sequence. The book of life was this huge volume. Before, only in the last 4 years or so, 5 years, have we come to realize that the differences between us in our volumes are not just individual letters that are different, but actually whole paragraphs and sentences and even chapters can be deleted or duplicated. And that shows great promise. Okay. So what is all this genetics telling us about function? Well, those genes we've identified, a lot of them affect the function of the synapse. That is, how neurons communicate with one another. It's not coincidence. Okay? So those individuals where we found some genetic changes, and recently in the last 2 years, a number of proteins that mediate the adhesion between different neurons have been identified. There's also a lot of information about translational control. Remember I told you that how you get this information, you have a copy of the mRNA, and then you translate it. As it turns out, this process is very important in learning and memory. So there are messages stored in neurons that aren't translated, and when there's a lot of activity, then they get translated. Activity-dependent molecular changes that underlie learning and memory. Now look at some of the genes that are involved in controlling translation. PTEN, TSC1 and 2, the fragile X proteins. Not a coincidence. I have a few more moments I want to talk about this topic. Evaluating media reports. The amount of information that comes at us, is bewildering. And I think it was about 6 months ago that there was a report in the Huffington Post about mitochondrial disorders and vaccine interaction. I actually dived into that literature as I found it. And I can tell you that this report was incredibly biased and misleading and troubling. And it's an example of how a little information can be really dangerous, including for Mr. Kirby, who, in my view, has been overtly irresponsible. So, how do you evaluate all this stuff? And we ask ourselves that question too. Well, this is kind of what I look at. What is the background and affiliation of the authors? Has been replicated by many independent investigators. And is the data strong enough to support a consensus? That is what we need in the end to really understand it. So what's the current state? We have a difficult truth, which is we cannot provide these answers. So sometimes we're in this very unfortunate position of a hypothesis comes up and we in academia say, no, that's not it. Oh, there's another one. That's not it. It is very unsatisfying to everyone, especially, of course, the parents. And, of course, in the absence of clear understanding, theories, most of them wrong, abound. And we just have to be prepared for that, sort through them rationally as best we can. But I do want to be hopeful, because one great thing that's come from all the media attention and all the family involvement across the country is terrific people and resources are coming to study this very important problem. I also want to end with a little bit of some hopeful findings. You often hear that this is a disorder of development, that the instructions that set up the assembly of the nervous system, that's where things are going awry. And so the logical question is, well, how can we fix it? Even if we can diagnose it, what do we do about it? Well, there's some data that is very promising using these models, and the model of Rett syndrome in particular, that when you put interventions in the adult after the nervous system has assembled, you still get correction. That is very hopeful. That says that maybe we should not be just looking at these as purely developmental disorders. So the models we've created, we have reason to think that intervening after the nervous system has been assembled can do something positive. To me, that's incredibly encouraging. I'm past stop. I don't know what the next sign is. This sign says stop. The next one probably says get the H off the stage. So I will close and acknowledge. So this is just the gang that I work with and collaborate with to give you some semblance of, it takes a lot of people to do this and a lot of collaboration, and that's what we're trying to do. And I want to thank you for your attention.